So I think this is what everyone asks all the time. What clustering method is best for my data? And my answer is always depends on the data. There's not one straight answer. So it seems like graph-based clustering works best, but there's a bit of a disclaimer there, because now we're using t-SNE and UMAP for visualization. And then, of course, the graph-based clustering agrees the best with t-SNE and UMAP because it's built on the same idea. So then we're assuming that the t-SNE and UMAP, my throat is really bad today. Sorry. We're assuming then that the t-SNE and UMAP and the graph is what best represents our data. And but I think from experience working, it seems to work really well. I think you get good biology out of it, and that's really why people are using these tools. There's not really, and in a lot of evaluation, you find the correct cell types and so on. So it is the gold standard now to use graphs in single cell data. But we'll see in the future if someone comes up with something new. Of course, I think it's good to try a few different methods. If you always get the same clustering result, or more or less the same, you know that it's really robust and you can trust it. And if we talk about bootstrapping again, having good bootstrap values is not always either a case where you say this is the best clustering, because it can be consistently wrong every time you run it. It doesn't mean that it's the best way of capturing biology. And then, of course, these like hierarchical clustering and so on, or k-means clustering works really well if you have a very simple data set, if you only have 2 cell types or 3 cell types in your data set. But it's when you're starting with these bigger projects where you have a tissue with 10 or 20 different cell types in them, it's really hard to represent the relationship between all these different cell types without using graphs, basically. So there has been a few papers trying to evaluate different clustering methods. It's always an issue of what to use for the gold standard there. The ideal is to have fact sorted cells, of course, where you know their identity, but otherwise you have to rely on annotation from one study and then compare between them. But you can easily evaluate how consistent different methods are, but to evaluate what is best is a very hard thing to do, I would say. But this is quite a good paper to look at, I would say, if you really want to dig into this. So what they did, they also tested 3 different ways of selecting variable genes. And either just filtering on highly expressed genes, highly variable genes, or using M3 drop. And then they have a bunch of different data sets and a bunch of different methods. In their test, so I should say that the dark colors and high AR value, it means that you have good clustering, and the yellow ones, then it performed really bad. They also did mixing of equal proportions of cells and so on in these different samples. Clearly using the M3 dropped didn't work so well. And clearly, like basically Seurat and SC3 and Monocle, these methods out here seems to perform the best when it comes to clustering. And here is basically a histogram with the ground truth as they defined it, and Seurat is closest to the ground truth. So it seems like using this graph-based SNN clustering, in this case they were using Louvain clustering, works really well. So it is the method I would recommend you to start with. And as you can see, here is a list of what people are like, what type of dimensionality reduction these different methods are starting with. Where PCA here in dark, pink is the most popular, what kind of clustering method, and what kind of input you have to the data. So there's a lot of different ways of doing it, but clearly this graph-based, and this was, what year was it? 2018. So at that time, there were a few papers with the graph-based methods. Most of these other ones here are not based on graphs. It's actually only Seurat here that's based on graphs. But then we come to another very important question. How many clusters do you really have in your data? You can increase resolution, or you can cut your dendrogram as low as possible, but there's no real answer to how many clusters do you have in your data set. It really takes knowing the biology of that data to make some reasonable decisions. But I think one thing to look at is to do differential expression, because if you split your graph into quite a lot of clusters, and you have like neighboring clusters where you hardly have any significant differential expressed genes, maybe it's not a good idea to split them into individual clusters. There are some tools to do sort of automated predictions, but I don't think that works at all when I've tried it. And then always check back to your QC data. Is what you're splitting just between batches or for different quality of your cells instead of something biologically relevant? And one nice tool to look at this is actually this Clustree package. So what it does is basically, in this case, testing with different resolutions or different settings. This is in SE3. So if we increase resolution a bit, this one splits into 2 clusters. But if we increase even further, we get 3 clusters, but some of the cells in that cluster ended up in that cluster. And then as you go on, and I think especially if you get a lot of errors going back and forth in different directions, it feels like the clustering is not super consistent. But it's a good way to get an overview of what happens as you increase resolution. And then, of course, if you have a big data set with a lot of different cell types, it might be a good idea to take out one cell type of interest and start from scratch, more or less. Because when we do variable gene selection, we will get the genes that best separates all the data in the data set. When we run PCA, we have the principal components that separate all the cell types in the data set. So by going back to just one cluster or several clusters that are close by, as in this example, and rerun variable gene selection, PCA, graph construction, and cluster again, you might separate out subtypes better than you did in the full graph. And as I said, always go back to quality control data. So here is an example. It's quite an old project where you see clearly, I mean, so each square here is a cluster as defined by this algorithm. But you see clearly I have strong separation by donor here. We can look at gene detection here. Here is actually the known cell type from fact sorting. So we clearly have strong batch effects within our cluster still. We clearly have slight differences in quality between the clusters. Here is one example from a project. The first round of clustering I did, you see, here is percent protein coding genes that are detected. For instance, you have one really problematic cluster here. You have some clusters with very high mitochondrial content. So if you see these kind of patterns, think about what they mean. Is this a technical artifact? In this case, this cluster, for sure, I would go back and think about filtering, remove those cells and rerun clustering. Maybe also go back to percent mitochondria, redo some filtering there, and then rerun again my clustering.
Can I ask about the reclustering of a subgroup? Do you still keep the same clusters, but you separate more, or do you make wholly new clusters?
I would do both. So I would rerun my the whole pipeline. I would plot this new UMAP, and I would do new clustering, and I would plot both the old clustering and the new clustering side by side on the UMAP, and then see if they agree or not.
I think in how to describe it, like in an article, if you have already described that we have these clusters, and then you take out this, it's very difficult if you get completely new clusters that are not like agreed.
Yeah, but for instance, I mean, if we say that now this cluster 0 splits into 3 smaller clusters, I would probably say that this is cell type A, subtype A, B, and C, and then I would probably have them in different shades of red because then you can actually take that annotation back to this big one and plot these in different shades of red as subtypes of this one cluster.
Great.
Something like that, I think, would be what makes more sense.
Good.
Yeah, so always do this. Plot these QC stats per cluster. If it very different sizes, if there some cell type is much more metabolically active and you have higher mitochondria in that one, then it might be fine. So really understand what the clusters are, understand how these quality control measures relates back to biology, or is it a technical artifact? Also check batches. I think it's a very good idea to, once you have your clustering, here is another project where so the smallest cluster we have is pretty much just dominated by one batch. And when looking into it, it also turned out to be a low quality cluster, and it was clearly a technical artifact. But in some cases, you might have a unique cell population that's only present in one of your batches. So again, think what it means and then make decisions. So if we go back to where we started, really think about these things before you start. Be aware that the choices you make here will give different results. So really think about how to interpret it. And of course, validate the results you get. Ideally, if you find new cell types that you didn't know were there, you should be able to validate them in the lab with some other method. And as a conclusion also, in most cases, it's not super complicated. if you have distinct cell types, they're quite easy to distinguish. But within cell types, subtypes, or different states of cells, then it starts being complicated. And if you have the same, it agrees with the GSNI, the UMAP, with different clustering tools, then you can trust the results more than just one time when you ran clustering, you got really nice results, but the other 10 times you ran, you got completely different results, then I would be worried. And use the biological knowledge to really evaluate if you can trust what you see.